knowledge on nematode infestations in polyhouse cultivation and their management. I am a nematologist working at Indian Division of Nematology, Indian Agricultural Research Institute, ICAR-IARI in New Delhi. As you know, the polyhouses are increasing in the country, and the polyhouse cultivation is being seen in almost every state. So, but there are problems, and especially nematodes are one of the problems which almost every farmer is encountering. So, in this context, I would be speaking on the advantages of polyhouse cultivation, increasing area under polyhouses, the crops that are cultivated, the reasons for nematode proliferation in polyhouses, which nematodes are of concern to us, the cultivar variation and susceptibility in vegetable crops, nematode problems in flowering crops, management aspects, and challenges in polyhouse cultivation. Now, there are various advantages of growing a crop in polyhouse. First is that we can grow off-season. Off-season production of vegetables, cut flowers, and fruit crops. There is less dependency on climatic conditions, the abiotic stresses. The temperature and moisture are more or less consistent compared to the open house conditions. There is longer cropping period. So the crops can be grown from 4 to 6 months at, and the yield is 10 to 12 times higher than the outdoor cultivation, depending on the type of the greenhouse, the type of the crop, the environmental control facilities, etc. There is quality production, and mostly the crop is disease-free and genetically superior transplants. There is efficient utilization of water because it is the drip irrigation system, and efficient utilization of inputs like chemicals or biofertilizers or pesticides if at all they are used. And it is good for cultivating and maintenance of grafted plantlets and micro-propagated plantlets as well. And there's hardening of the tissue culture plants. Also, this polyhouse cultivation is advancing, and there are many private entrepreneurs who have taken up hydroponics, soilless culture, aeroponics, and other techniques. Who can, but it is of course cost intensive. But for most of the farmers, there are increased profits when they take up to polyhouse cultivation. Is the slide readable? Madam, is the slide readable?
Yes, it is. You can
Hello.
zoom it a
Yeah.
little more.
Okay. So this is the off-season vegetable production under low-cost polyhouse, and I have tried to show the profits which one can earn in after growing gherkins, cherry tomatoes, bitter gourds, or cucumbers. So you see the profits vary from 23,000 to 43,000 up in 1,000 square meter area. This is in season one, but as you move to seasons two, this profits are enhanced to 59,000 in cherry tomato, 50,000 in cucumber, 52,000 plus. So this is the reason, because it is highly remunerative, that farmers are taking up polyhouse cultivation. And in most of the countries, the area under vegetable crops is the maximum. And see, this is the total area, and about 115 countries have taken up polyhouse cultivation. China is of course leading, followed by Korea, Spain, Japan, Turkey, Italy, Mexico, Netherlands, France, and United States. So whether it is, I haven't shown some countries like Israel, Russia, because in polyhouse cultivation is being adopted in countries which are showing extremes of climates, like in Russia and Canada where there's extreme winter, in Israel where there's extreme summer, in Philippines where there's excessive rainfall. So polyhouse, these uncertainties, the polyhouse can overcome. So the cultivation under polyhouse is definitely preferred. In a country like India, where we have tropical wet conditions, tropical dry conditions, subtropical humid conditions, mountain climatic conditions also, and the average precipitation is just 1083 per mm per year. The temperature range is -13 to 51, and the arable land is also limited. gross irrigated area is limited. There also now the polyhouse cultivation is increasing. This the cultivation practices, it started polyhouse, first polyhouse was built up in by DRDO somewhere in 1990s in Ladakh region. And you see, as the years passed by from 2005 onwards, the area under polyhouse, it kept on increasing from 451 hectares, and presently it is about 30,000 hectares. 30,000 hectares, and almost every state has got polyhouses. And some states like Haryana are giving incentives also to the farmers at subsidized rates to erect the polyhouses. Now, what are the crops that can be taken up in a polyhouse cultivation? Most of the polyhouses are growing vegetables, which include tomato, capsicum, and cucumbers. These are the top priorities in our country. Besides, some have taken up strawberries. Then there are orchids also in northeastern states. Then there are also cut flowers, because cut flowers are highly remunerated. They fetch a high price because of the quality produced. All this is fine, but then all these crops, they are encountering the problems of nematodes which are present in the soil, and they are a hidden enemy. We cannot see them with the naked eye, but there you see this is a tomato crop which was grown somewhere in the soil, but when you harvest it in March, you can see the heavy galls on the roots. And in September we see it like this. There are no symptoms at all. But when you harvest it in March, you can see the heavy galls on the roots. So these are tumorous like growths. And when the farmer comes to know that there's something wrong in his soil, and he will understand that there's some problem, and he needs to go for some treatment. Many times the density of the nematodes in the soil is so high that the germination is very poor. For example, in this slide, in this picture, I've tried to show that germination of muskmelon is so poor because of the high nematode density. And the only symptom of this root knot nematode is that it produces galls below, heavy galls. Above ground, many a time, yellowing of the plants may be observed, or early wilting may be seen under water stress. But in polyhouse, there is hardly any water stress because of the drip irrigation system. So you don't see plants wilting also. And because there's intensive application of biofertilizers and chemicals, so you don't see yellowing also. So the symptom remains hidden, but definitely the plant is under biotic stress, and the quality of the fruit is affected. Sometimes even the yield may not be significantly reduced. It may be reduced, but it may not be significant. The quality of the produce is definitely affected. So this is the kind of galls you see. And when you see stain the root system, you can see the number of nematodes. These are adult females of the nematode which you can see inside the root, and this is on the outside. Here also you can see these roots on the outside. So when we have done sampling in polyhouses, we have seen the populations are sometimes from undetectable levels to as high as 80 nematodes, infective juveniles, per cc of soil. In 1 gram of soil, 1 cc of soil, we can find 80 infective juveniles, which is a very high number. So we have been going to different polyhouses, and we have been collecting samples, and we have seen that almost all crops, cucurbits especially, are highly infected with this nematode. Now this nematode will produce galling on the roots, but another symptom is that it predisposes the crop to other pathogens, especially the fungal pathogens. Now, sometimes fungal pathogens are just in they are present in the soil, but they may not invade. But what happens when nematode is present? Nematode acts like a wounding agent because it has got a stylet, like your needle-like, structure in its mouth with which it punctures the root system. It causes mechanical, wounds on the roots, and when it causes wounds, the nematode, for its feeding, it provides a mode of entry to the fungi which are present. So the fungi, they also enter. Secondly, they act as rhizosphere modifiers because when nematode is feeding in the root, they modify the secretions which are there in the rhizosphere, because of which also the many pathogenic fungi or bacteria may proliferate. And third is they cause breakdown of resistance. Some plants may be resistant to fusarium wilt or pythium or other pathogenic microbes. But what happens is when nematode is feeding, it breaks the endodermal layer of the in the plant, which is responsible for secretion of phenols and responsible for resistance. So when this endodermal layer is broken down, then the resistance is also broken down, and there is invasion of vascular pathogens. So besides the nematode damaging, it predisposes the plant to other fungal and bacterial pathogens. That is why it is very important to take care of them at an early stage and to manage them. Besides these vegetable crops which are most prone to these uh, root knot nematodes, this is even the cut flowers, the gerberas and the carnations which are grown, they also are being affected by these nematodes. And in Europe they have accounted 40 to 60% loss due to this nematode. In India, about the losses are about 26 to 30%. I hope my slide is moving, madam.
Yeah, it is
So
moving.
yeah, so you see, we have there are various reports of nematode infestation in various crops in different parts of India and outside, especially in Haryana. You can see, besides the root knot nematode, there are other nematodes like Pratylincus, Rotilinculus, Hafnilamis. Some are ectoparasites, some are semi-endoparasites, some are endoparasites, etc. So you see, there are various reports of these nematodes from different polyhouses. But if you see the table carefully, you see that Mylidogynous species, that is incognita or sometimes javanica, it is invariably present and reported in from all polyhouses. Even in Spain, you see javanica, M. incognita, M. marinaria, all these have been reported and are a serious problem in the polyhouse cultivation. Where do these nematodes come from? Now, it is very important to understand that what is the source of the infection or the nematode. So most important is the contaminated soil or soil mixture, which is used as a component of the growing medium. Sometimes we are growing our saplings in in the growth medium, in the coconut peat, or the soil mixture is already contaminated. So we need to check if nematodes are present. Sometimes we are procuring the nursery from outside. So the place where nursery was grown was infested with the nematodes, and so the nursery is carrying the nematode along with it. The nematodes are already penetrated inside the root system. And third factor is the monocropping, because normally in polyhouses we grow the same crop of, first month, second month, so the duration of the crop is also long, and sometimes they are all susceptible to the nematode. So monocropping is something which helps in the build up of the nematode densities in the polyhouses. So nematode management must be considered very early and timely, because once the nematodes are introduced, it is difficult to manage them. It is like cancer. If cancer is detected on time, then you have the remedy for it and it can be treated. But if it is late, then it spreads. So same is the case with this nematode. Now it is I would give you some description of how the nematode proliferates in the soil. So it is the the eggs or the juveniles, the female is there which is laying eggs. Now I would like to start from the eggs, like from the egg, it is the second stage juvenile which comes out of the eggs. And the second stage juvenile, it penetrates the root system. So if we have washed the soil sample or extract nematodes, we only get the second stage in case of root knot nematodes. And here I have tried to show you the emergence of the second stage from the egg. This is the live picture, and this is the egg where the nematode is in a coiled state. Now, once there is emergence of the juveniles in the soil system, then they will penetrate the root system, and further developments, third male, female, and other, the whole development will take place after once the nematode starts feeding in the susceptible crop, and it then develops into a globose female like this. So it is a globose female, and then it will develop after about 25 days. It develops egg mass, and this egg mass will be filled with eggs which may vary from 300 to 500 in number. So the whole life cycle is completed in about 25 to 30 days at optimum temperature. So if the temperature goes below 8 degree, or if it goes above 37 degrees, then the further development of the nematode is arrested. But this does not happen in polyhouses. The temperatures neither go below 8, nor do they, when the crop is there, they do not, the soil temperatures do not shoot up above 37. So this is the reason that nematode is able to complete 7 to 8 life cycles, in the cultivars, whether it is cucumber, tomato, or capsicum. Another favorable, you see, I have shown you the soil temperature difference in polyhouse as compared to the open field condition. There is one solid line which is showing the temperatures in the polyhouse, and a dotted line which is showing the temperatures in the open field. So on an average, there is a difference of about 4 to 5 degrees, if we have recorded the temperatures from January to December. So throughout the year, the temperatures are conducive for the nematode development. Now, I would reiterate, so there is intensive cultivation, and also the varieties which are grown in polyhouses are at least 4 to 5 months for long in duration. So that enables the nematode to complete multiple life cycles. There are favorable moisture conditions because there is drip irrigation. So the nematodes keep proliferating. The temperatures are very favorable. There is depletion of the natural enemies because there's intensive inputs, large number of. So normally we do not find natural enemies in the soil. Besides this, root knot nematode, I've tried to show pictures of another semi-endoparasite. This is Rhotilinchus reniformis, this is Pratylincus, and then there are some ectoparasites also which are present. But these ectoparasites may be just feeding from outside and may not cause much harm. It is the endoparasites and the semi-endoparasites shown above which are of more, damaging in nature. Another aspect is that not all the cultivars are equally susceptible. Like for example, in tomato, there's a variety called GS600. See the kind of galling it is showing, heavy galling, tumorous growth, very large size galls. There is another variety in capsicum Indra, which is showing very small galls. So the galling, the size of the galls also is highly variable. This we have observed in capsicum also, in tomato also, in cucumber. The size of the galls are not very variable. So this was an observation which we made way back in 2013-14, that in tomato we saw that GS600 was heavily susceptible, highly susceptible. It is showing 300 juveniles, in 200 cc. So and or also Rhotilinchus reniformis was there, but cherry tomato was relatively less susceptible to root knot nematode. When we did sampling in the month of November, in January again, and then in March. Same way in Capsica, Indra was more susceptible. Swarna, Natasha, Orabili, they were less susceptible. In cucumber, the variety Kian was more susceptible. When we have done, numerically, we have counted the nematodes in the soil system. So after this observation, we tried to see the cucumber, because these days large number of farmers are approaching us, who are growing cucumber because it is fetching them a good price, and they want to know if there is any cultivar which is resistant or relatively less susceptible to the nematode. So in due to this reason, we did the screening of about 12 commercial cultivars of cucumber, which is a parthenocarpic type in protected cultivation, and it flowers in 25 to 30 days, and the first harvest can be taken up just in after a month or so, and the production is about 300 to 400 tons per hectare in most of these commercial cultivars. Now our initial approach was, we can also in the laboratory we can say, test whether a cultivar is susceptible to the nematode or not. So there is a assay called pluronic gel bioassay. Pluronic gel is it is a polymer, and it is a transparent polymer which is in a liquid state, in a semi-liquid state, in the at room temperature, and so the nematodes can move towards in the gel, and they can be even observed. What is their behavior towards the root? Are they approaching the root system? So under the microscope, it is a transparent gel, and the roots can, it is non-toxic to the roots. And using this fluoronic gel, we have assayed different cultivars of cucumber, and we have taken the rootlets, about 3 to 4 days old saplings of cucumber, and we have placed them on the fluoronic gel, and the tip of these have been exposed to the nematodes, and then their behavior of the nematodes and the penetration of these nematodes in the root tips have been observed. So when this kind of bioassay was conducted, we saw that all our cultivars, whichever cultivars we have used, all showed the penetration. You can see these, red colored stained nematodes, which you can see inside the roots. So all the cultivars were susceptible, only that some of the cultivars were more susceptible than the others. For example, there is a variety called Romeo which showed significantly higher number of juveniles than Eviva or others. So but infection level was relatively very, very, very high. So among 50 nematodes, under laboratory conditions, this was the infection level. Then we also sowed these cultivars in soil system, and we have grown the crop for about 3 months and seen the galling, because galling is also indicative of infection. So we have tried to see which cultivar is showing more galls, then it is more susceptible. So Romeo showed heavy galling, Dharwad green showed heavy galling, Minni, Malini heavy galling, but Captain Star was one which was relatively showed less galling, so less susceptible to the nematode. So this is the way these roots they look like. And because I can't show the close up, so but there are galls in all the roots. So all the galls, so in Romeo you can see the galls, heavy galling here. So all these crops were showing, all these cultivars were showing heavy galling. Now if this is the case, that most of the cucumber cultivars are, and same is that with tomato also, our trials are ongoing with tomato as well. If this is the case, then how do we manage if nematode is a problem? The first and the foremost is that we have to use phytosanitary measures. Our soil should initially be free of the nematode. It should not have even very low populations of nematodes. So phytosanitary measures, soil should be free, seedlings should be free, saplings should be free, and our shoes should not come from infested polyhouses. Our tractors should not go in polyhouses which are infested and then go to uninfested polyhouses. So phytosanitary measures are of utmost importance. Then we have many biocontrol agents which can be repeatedly used. One time application is never enough in polyhouses. So after every 3 months, repeated application is required. Then if still our problem is not solved, then we can go for fumigation. There are some nematicides which are available in the market, but as you all know that chemical pesticides are very, very harmful to the environment, to human beings, and they cause various problems. They may cause skin irritation, they may cause eye problems, they may cause fetal deformities. So we do not recommend fumigants. But some farmers, because they are having the heavy infestation, they want to go for chemicals, then I will tell you the fumigant options, but we do not recommend them in general. Also, there's a lot of research growing on grafting techniques to produce nematode resistant cultivars, but these are just on experimental basis. The trials are ongoing. So as I said, the inspection of the field before you erect a poly house. See, this poly house is being constructed, and they this is the we are collecting samples from them, from various places, and we make a composite sample, bring the sample to the lab, do the extraction, examine the suspension under the microscope to see if root knot is there. See, soil will never be free of nematodes, but does it have nematodes which are which could be harmful? So we have to, you need a nematologist to identify that this is the root knot nematode or juvenile of a root knot nematode, because there are many nematodes which are even beneficial to the soil, our crops. They cause, they help in the process of mineralization, so they are not harming our plants. If they are present in high numbers, no problem. But if you have a parasitic nematode which has got this kind of a stylet, then you have to consult a nematologist. And then you at the timely application of a biocontrol agent, or a fumigant, or whatever chemical you want to adopt, you need to decide on that. So this is regarding the various work which has been done in different parts of the world, in Egypt, Iran, Iraq, India, how they have been using these biocontrol agents, and they have got success. You see, in Egypt they have been using, Iraq they have been using trichoderma, purpureocilium, India also Pseudomonas fluorescens, purpureocilium, trichoderma, etc. So these are the common biocontrol agents which are available in the market with, many companies are selling them. So you can use a quality product for managing these nematodes. So also there are countries like in Spain and Switzerland, they have used pesticides, fumigants like fluncylphone or flupiram. Besides, biocontrol agents, soil solarization, covering the soil with 100 micron transparent polythene sheet for about 6 to 7 weeks. That also builds up the temperature inside and it kills the nematode. And this is a chemical free treatment. The only thing is that you ought to buy this polythene, and then degradation of polythene is an issue. But otherwise, summer solarization, which it is called, or polythene mulching, is highly effective in managing these nematodes. So these are the, I've tried to show the various formulations, like especially Pseudomonas fluorescens, BioShield, Spurge, Pseudoseal. These are the various products which are available, and the various companies which are selling them, and you can see them on the net. You can discuss from, ask the nematologist which product is good and which have been tried and tested, because there are many formulations which are also spurious. So it is better to buy a good formulation from a university product is has definitely a better quality, but it is not always so. Some of the companies like Panacea Biotech and all, they are producing good quality products. So you can purchase a product like this and apply it in the soil. Pre-plant application is recommended. So these are some of the products which are available at international level also, which can be used in drip irrigation. So we have Purpureosylium lilacinus, a vegetable powder. These are products, and then PL Gold is available for tomato. Bacillus firmus is available, and liquid formulations called Bio Yield of Penny Bacillus macerans, etc. Nimex, Bacillus, nematicidal Bacillus species is available, which can be applied. So now application part is two-way. First is the nursery application, and then is the field application. So when you're preparing the nursery, then in these trays, the nursery trays, you have normally use the cocoa peat. So this cocoa peat can be enriched with the biopesticide, whether it is trichoderma, pseudomonas, and it can be thoroughly mixed, and then it can be filled. And the nursery which is raised in this way will be free of the nematode. So this precaution can be taken, whether it is to ensure that the seedlings do not carry the nematode infection. Then what we have done, we have also tried Pseudomonas fluorescens. So when the beds are being prepared, we have applied Pseudomonas fluorescens in the rows where planting has to be taken place, and we found that our nematicidal isolate was very very effective and declining the population. So we have applied it in the rows, so you don't have to apply in the whole field. So then we have, after 4 months, we have seen that the our plants were free of the galls. So there was no nematode. So, and we have compared this with neem cake. So neem cake also, there was some increase, but see, in the first month the population was low, second month the population was low, and if you see in PF and neem the population was low. So Pseudomonas alone was also very effective till at least 3 months, till at least 3 months of sowing. But sorry, but after that, after 3 months there was a surge in the population. So what we recommend is that every third month also, if you can apply a biopesticide like Pseudomonas fluorescens, it will keep the population densities very low, below the economic threshold level. So your crop would be safe. Now, what is the how do these biocontrol agents they act? Like I said, Pasilomyces lilacinus. These they parasitize the eggs. So even if the there is egg mass coming out of the root system, these Pasilomyces lilacinus, now it is called Purpureosylium lilacinum. It will invade the eggs, and it will kill the eggs. So there is parasitization of the eggs, and also there will be some products which are released, chemical toxic substances released by the fungus, which will kill the eggs. So the juveniles will not come out of these eggs, and it will protect your crop. Same is the case with Paponia chlamydosporia also. So these fungi which have been identified, they are very good proliferators in the soil system. That is why their products are available in the market. Now there are some predators also like arthropods and all, but they may not be that effective, because when there's no crop, the arthropods and all, they don't survive. So Pseudomyces and Paponia, they're good products. Same way Pseudomonas fluorescens is also very good product. There is another bacterium called Pasteuria penetrans, which can be introduced in the soil, but this unfortunately there's no formulation available for this bacterium so far, but it is a very good parasite of the nematode. And then we, of course, we have trichoderma, and which is also effective against pathogenic fungi. So there sometimes the dual effect on nematodes as well as pathogenic. So this is the picture to show how Perpuricillium it parasitizes the eggs, because the eggs are there outside, which are exposed, out on the exposed on outside the root system. So if you add Perpuricillium in the soil, it will parasitize. So these are the healthy eggs which are with the nematode, and this is the eggs which are with the fungus. So this is how the fungus looks like. It has got conidiation or sporulation like penicillium. Same way, you have this Poconia chlamydosporia. See, this is a scanning electron micrograph to show how the fungus proliferates on the eggs. It is this parasitization inside the egg, and then these are the resting spores. So these are Poconia is a very good biocontrol agent. Same way, trichoderma is also there. Similarly, some of the isolates parasitize the eggs also, and trichodermas are very good competitors in the soil. Now, this is all about biocontrol agents, but sometimes the you haven't planted and the farmer wants to go for a quick, remedy, because biocontrol agent will reduce the population. They won't eradicate. So nematodes. So sometimes the farmer says to chemical bataiye. So in that case, sometimes the farmers, before planting, they will drench their soil with 40% formaldehyde. Now this formaldehyde also kills the nematodes, and you have to cover the soil with polythene for at least 2 weeks, and then uncover it and leave it for 7 days so that all the fumes will evaporate, and then go for sowing or transplanting. This is being adopted by some of the farmers, but it is not always effective. There is another chemical called methampsodium sodium or VAPM, which is available by Sudama Chemicals or Adama, and it is a general biocide. It will kill all pathogenic organisms, including nematodes, also pests, and but it is highly toxic in nature. So when you are applying, you should wear mask, you should use gloves, and then you should apply only where required. No need to apply in the planting rows. You can apply them, you can drench the planting rows with the chemical, then cover the rows with the soil, and then cover it with a polythene, leave it for 2 weeks, and nobody should enter that polyhouse for 2 weeks. After that, one has to open it, leave it for about 7 to 10 days, and then you can plant. But I may tell you that it is a complete biocide. It will kill even the beneficial organisms. So should be used only when it is of utmost importance. In the market, we have fluoxanthone also. So this can also be used. This is recommended for some crops. So it can be used again in the planting rows only also. Okay. So this is an expensive pesticide, about 2,500 per kilo. So it can be recommended for tomato, capsicum, and cucumber. So this is again recommended at twice. So first at the time of sowing or transplanting, and then again the second application is recommended after 25 days. So dose is 1.5 gram per plant. So you can also do, if at all you have to use a chemical, please do spot application. And it is also recommended for cucumber, okra, tomato, capsicum. This is nemates can be utilized. There's another chemical called vellum prime, which is becoming very popular among the farmers. Vellum prime is actually a fungicide, flupirum, and it is available in the market at about 7,500 per liter. So, of course, the dose applied is very less, but it is recommended for vegetable crops, strawberries, and cucurbits. So, it can also be used in drip irrigation system, but the results have been variable. Now, besides these pesticides, I would also suggest one more method of nematode management, which is not much used. It is called biofumigation. So, what happens? There are many cruciferous plants and waste material. If you have cruciferous plants like brassicas or cabbage leaves or cauliflower leaves, biomass, which is a waste material, then that waste material can be finely chopped and it can be incorporated in the rows, and then it can be covered with the soil and covered with like polythene mulch and left, because these organic matter, these biomass will also release toxic chemicals called glycosides, and these release chemicals MIC called methyl isothiocyanate, which is also nematicide. So it is like a fumigant, it acts, but this has to be done when the when you have to uproot the plants. If the field is infested, you realize that there is nematode infection before you plant the next one, then you can do this. The it should be free of the crop system. And then here also you have to cover, because this degradation of this, these leaves, they will release the gas, and because it is in gaseous nature, the covering is very very important. If you don't cover it with a mulch, then all that gas will evaporate and the effect won't be seen. So biofumigation is also effective. So 2 to 3 tons per hectare needs to be applied. So the constraint is the availability of this, biomass of cruciferous plants at the right time. This is a constraint. And of course, as I said, that many plants are being explored for grafting purpose, so that we can come out with resistant plants which were not susceptible. So root knot resistant tomato bichians are being tested for this purpose, so that in future we may be able to have plants which are resistant. So there's no infection. Even if nematode is present, there'll be no infection. Of course, that will also have limitations because the resistance is temperature governed. So if temperature shoots up, sometimes the resistance is broken down. It will also depend on the population densities of the nematode. So there will be limitations to this approach also. However, I will again say that safe methods should be used for nematode management. There are many research gaps in these areas. For example, we are not aware that if there are different races of root knot nematode which may be prevalent in different polyhouses, maybe some races are more aggressive than the others. Then the vertical distribution of the nematodes in polyhouses needs to be studied. How deep? Because these commercial cultivars are growing up to even sometimes 3 feet deep. So the nematode also travels that deep. That is why it becomes very difficult to kill, and we have to use fumigants at times. And we have to identify nematode tolerant or resistant cultivars for grafting purpose. We also need to keep in mind that there are other nematodes, like another species called Mylidogyne enterolobi, which was earlier not prevalent in the country, but it has now been introduced from outside, and it could be a serious threat. Even Rhotiluncus reniformis could be a serious threat. It is presently not that much prevalent, but it could be a serious threat in the future. And we also can explore the possibilities of using this endoparasitic bacterium bacteria, which is a which can is able to survive in the soil and resist high temperature fluctuations. So this could be a very good approach if bacteria could be used. Of course, so far we do not have a formulation. It has to multiply along with the nematode, and it can be utilized for suppressing the nematode population in polyhouses. So with that, I would, end my talk here. And this was my last slide. I would thank all of you for your patient hearing, and I'm ready for the questions, if any.
Thank you, Dr. Anju Kamra, for a very detailed presentation. I'll take up the questions now.
Yeah.
Okay, the first question is,
Yeah. And could you be a little louder, please?
yeah, why does polyhouse cultivation increase nematodes compared to open field cultivation?
Yes, as I told, that the favorable conditions of temperature and moisture are prevalent throughout the year, almost throughout the year, except the month of in the northern conditions, May and June. Next, in areas like Rajasthan and Haryana, May and June it doesn't support any crop. But otherwise, the temperature is very conducive. Even in winters, the temperature becomes conducive. Then, because of the drip irrigation system, the moisture conditions are conducive. And thirdly, the varieties which are grown, they grow for 3 to 4 months. So it enables the nematode to multiply, one of several complete several generations. That is the reason.
Okay. Next question is again related to the same. How big a problem are nematode in open field cultivation?
Sorry, how is it important?
How big a problem are nematodes in open field cultivation?
Yes, in open field also the problem exists. But what happens is that the farmer goes for crop rotation or non-host crop. He will grow the non-host crop. In polyhouses, we have limited options. to not to grow a crop which is susceptible. So that is the reason. In open field also, the problems are varied. It is not just incognita and javanica. We have Milidogene, graminicola is there in rice wheat cropping system. We have Milidogene indica. We have interlobi. Many problems are there in open field conditions. But in polyhouses, it is mainly the incognita and javanica which are problematic.
Okay. And next question is, does nematodes means disease causing organism in the soil?
Yes, I said that they predispose the crops to other pathogens like fusarium, pythium, other vascular pathogens. So they can be called disease-causing agents. Otherwise, they are pests. They are parasitic on the plants. They complete their life cycle on the plant.
Okay. And next question is, what are some basic measures a polyhouse farmer can adopt to reduce the nematode problem?
Yeah, as I said, the first and the foremost is that when he is planting in a polyhouse or even before erecting a polyhouse, he should make sure that his soil is free of the nematode. So even very low population densities can become very high later on. So the initial population density is of importance. So you get your soil tested from a KVK or any university lab or whatever place is nearest. So make sure that your soil is free of the nematode. That is the first and the foremost. Then your saplings, what you are planting, make ensure that they are not infected. Examine them, get them tested before you plant them. If you cannot get them tested, at least you apply a biocontrol agent in the soil where they are being grown. Procure them from a place where you are sure. We see there is a private nursery in Andhra Pradesh which has led to the spread of Milidogenic graminicola like anything. I will not name the nursery. It's a private nursery which has spread the nematode because the place where it was raising its nurseries was heavily infested with root knot. So these initial measures are of very 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 important. And these days, they are the National Horticulture Board. They have made it mandatory that if any farmer or entrepreneur he wants to build a polyhouse, he has to have a certificate to that effect that his soil is free of the nematode. So our All India Coordinated Research Project, we have the headquarters at Delhi and centers all over India. They accept the samples and they examine the samples and make they certify if it is nematode free, or they tell you whether the infection is there and what level of infection is there. So you can approach the ECRIP center also.
Okay. And next question is nematode a big problem in greenhouse flower growing?
Yes, in flowering crops also it is a problem, but the reports and the most of the reports are from outside India, from Europe and other. I'm sure the gerberas are also, and the chrysanthemums where are they are grown in India also. IIHR has some reports of polyhouse infections, uh, the nematode infections in polyhouses. But I don't have a detail. I haven't worked with it, and I didn't have the data for that. But of course, there are a few reports from India as well because gerberas are susceptible, chrysanthemums are susceptible. So the cut flowers are susceptible to the nematode, and the losses are accounted in Europe and elsewhere. So there's a need. We need to be cautious because if a poly house has some vegetable crop growing in it, and later on he switches over to cut flowers, he has to be very careful.
Okay. Okay. And next question is, how much awareness do you think there is among polyhouse farmers in India about this nematode problem?
See, initially the awareness wasn't much, but now, as you know, with the social media coming up, there are so many WhatsApp groups, and I came to know that there are farmers who are in polyhouses who have polyhouses. They have a different WhatsApp group. So through all the social media, the awareness has definitely increased. To farmers, if we could not, scientists could not approach, at least through the social media, they are very much aware. And the number of calls we get from polyhouse farmers, it indicates that the awareness is there, definitely.
Okay, good to know about this. And next question is there is a large scale nematodes problem in a polyhouse, what options a farmer has at that point if there is a large scale nematodes problem in a polyhouse?
Okay. So large scale problem means I would always say that avoid a pesticide. Depends what crop he's growing. So I would we would always say, you can apply if the population densities are more than 50 or 60 per cc of soil. So get your soil examined. If it is very high, you can go for a pesticide, you can go for methampsodium as well, but you have to be very very careful. The application part is very careful, so it should not cause any harm to the applicators, to the people who are applying. So awareness in that aspect is very important. If you are using a pesticide, cover your mouth and your hands, and nobody should enter the polyhouse once he has applied a fumigant. So if sometimes the densities are so high that a biocontrol agent may not affect. So but for application of a fumigant there should be no crop. So depends what stage. Now if the problem has been detected at a later stage, then you can definitely apply, drip irrigation. You can apply liquid formulations of Pseudomonas fluorescens. The cost would be rather less. And every third month, I would say, you give a second application, then third application. You will give repeated application. It will it will maintain the population at low levels. So
Okay,
it is the option is always the decision making is done by the farmer only. We can only tell him the pros and cons.
okay. And next question is chemical fumigation the only way to get rid of nematodes? Are there any organic methods to get rid of nematodes?
No, as I mentioned in my talk also, that soil solarization is a chemical-free method, and these days, the ICAR-IARI scientists they are recommending soil solarization in polyhouses which are free of the crop. So you use 100 micron transparent polythene sheet, and it is spread on the soil after moistening the soil. It is left for at least 4 to 5 weeks, and the nematode population just drastically reduces. It hardly any nematode can be found. So it is a
Oh,
chemical free method. And you can amend this soil during solarization with the biofumigant, like, any biomass, like these cruciferous waste. You can amend it with, neem cake kernels, if it is of good quality. You can amend the soil with these, followed by solarization. So these are safer methods and they're effective also.
okay. And next question is, Madam, could you please tell us about your own education and research background?
Yeah. I have done, I'm a graduate from Delhi University in botany. Thereafter, I did my master's from IRI in Division of Nematology, IRI, where I pursued my PhD. I joined here as a scientist in 1993. So I have been guiding MSc and PhD students and held several projects during this time. And I have worked mainly on biocontrol agents of nematodes, but I have also had a project on beneficial nematodes. Nematodes which are not which are present in the soil and which have a role to play in nutrient mineralization. So they are very high in places where high organic matter is there. So that is a different area of research. So this is my, and presently also I'm working on some biocontrol agents and on similar aspects. So
Okay,
I'm a principal scientist at present and I have guided about 10 students. That's
okay.
about me.
So, what made you? Next question is again related to the same thing. What made you take an interest in this particular topic of nematode problem in polyhouses?
Actually, it was the interest of the farmers. The problem was emerging, and the number of farmers which were approaching us during our krishi melas and others, we were getting phone calls. It was the problem at the field level that we took up, that this should be, because the farmers are facing this problem. It is an emerging problem. We should work on it, and we are still working on evaluating the various methods of management, because it's an emerging problem.
Okay, okay. So the last, yeah, we have an appreciation message from one of our participants, and he says, "Excellent talk packed with information and research findings," and he thanks you for this. detailed
Thank you so
presentation.
much.
Yeah,
Thank you
you're
so much.
welcome.
Thank you.
So yeah, so now we have come to end of question round. Agricultureinformation.com likes to thank Dr. Anju Kamra for a very detailed presentation, and we also like to thank all the participants for joining this meeting. The meeting will now be closed.
Thank you so much. Thank you, Dr. Murthy.
You're most welcome, ma'am. Thank you.